From the most trusted names in local news, KDPG Sunday Edition.
And good morning. Welcome to the KDPG Sunday Edition. I'm Rick Dayton. In history books around the world, kids learn about the name of Jonas Salk. It was 1947. The University of Pittsburgh recruited Salk to develop a virus program at Pitt. It was his team that worked for years to develop a vaccine for polio. They succeeded, and it created a legacy for Pitt as a place for life-saving medical breakthroughs. That legacy at the University of Pittsburgh is being talked about once again as Pitt's Center for Vaccine Research enters the fray against another worldwide epidemic, the novel coronavirus. Coronaviruses, they're nothing new, but this particular one is. First identified late last year in Wuhan, China, and now Pitt researchers are in the process of obtaining samples from the novel coronavirus with the idea of developing a vaccine. Joining us this morning in the KDPG Sunday Edition is a man leading that mission, Dr. Paul Dupre, director of Pitt's Center for Vaccine Research, also the university's Jonas Salk Chair for Vaccine Research, and also on the program this morning, Jerry Micco, Senior Assistant Managing Editor of the Post-Examiner. Good to see you.
Good to see you, Rick.
And Doctor, thank you very much for being here today. We start with a show and tell, and it's a box that I want you to give us some sort of idea as to why this box has a lot of people in Pittsburgh, whether it's concerned or talking or questioning or wondering what's inside.
Well, this box, whenever we receive a virus from somewhere where there is an outbreak, this box is how we transport these viruses safely to Pittsburgh. So you'll see it's rather involved because there's layers and layers and layers. So as I unpack the box in which the viruses have arrived in Pittsburgh, you can see that inside the box is another box.
Okay.
So this is the styrofoam lid that keeps everything together. And then whenever the virus is coming, because it's a biological agent, there will be dry ice around this to keep it really, really cold because we don't want to get it damaged as it comes across the ocean or up from Atlanta in CDC.
Got it.
So we open the box within the box.
Box number 2, yeah.
And inside box number 2, we have another container, and this is a very special container.
Okay, I'm going to set this down. Go ahead.
Which is used to transport the agents around the world, and this happens all the time. Viruses move around the world in such containers often.
So if you see a container like that, you've got an idea of perhaps what's inside. Is that correct?
Well, we have a lot of documentation outside the box. Before anyone would open this box, people have to be trained for 2 hours to learn exactly how to package this up. So we don't just let random people send these things around the world.
So we've gone from one box into another, now to a cylinder.
So now we open the cylinder, and you can see that this is tough plastic. So this cylinder, inside the cylinder we have a bag. Inside the bag we have another piece of absorbent material. The absorbent material is there if, for whatever reason, something happened. It would leak up the virus, but the virus would still be present within this sealed bag, within a sealed box, within another box, within another box. You see the 3 layers?
Right.
And then whenever we open the bag and take out the material here.
The absorbents come out.
Absorbents come out,
right.
And then what you get to see, and remember, these are just demos, okay, but I made it look exactly like what we got in the lab. You can see that here we have a tiny little tube with 0.5 milliliter of liquid, and it's got the name of the virus in the tube. It tells us how many times it's been grown in the laboratory, and it tells us the date in which it was sent. And even at that, we also have an additional layer where we have this little sticky stuff on the top of the tube. So this is the protection and more protection and protection.
Right.
So that as soon as we take this off and we open the tube, if this was a real virus, that's how we would access this virus. And in this tube, there are 50 to 60 million virus particles in the tube.
In each tube, 50 to 60 million.
Yeah.
And you're looking at a population of Pittsburgh of, say, what, Jerry? 300? Yeah, I mean, the metro area.
Metro, 300,000 in the city.
And you start to do the math, and people say, why is this here? Do you understand the people's concern at home?
Absolutely.
Why would that, with those kind of numbers, be here? Why?
So The reason why we bring the viruses to laboratories like ours in the Centre for Vaccine Research is because they are purpose-built to receive such viruses from anywhere in the world. They have significant containment controls. They have significant air control. They have all of the individuals who are super well trained with other viruses which are very similar to this, so that they can safely receive it, safely unpack it, safely take it out of this little tube and put it into cells where we can propagate or grow that virus in the right laboratories, in the right place, with the right people who have all the skills to do the work. So why I brought you this was because it helps people understand a little bit about what comes. It's not volumes, it's not liters and liters of virus. It's these tiny little tubes, and then we, in those laboratories, which are purpose-built, safely grow it.
Pitt is one of, I think, a few places that are working on this. I think in the story that we had in the Post-Gazette, Dr. Dupree, you said, this is our part, this is our role, this is what we've been asked to do. But there are other places around the country or around the world that are also doing similar research?
Absolutely, and there are virologists all around the world who have been trained for this moment. The virus is circulating in China. It's reached many other parts of the world. We bring it to the laboratories to work with it safely, and we have many other colleagues in many other parts of the world, for example, colleagues in Switzerland, colleagues in Germany, colleagues in France, colleagues in Austria, colleagues in the Centre for Disease Control, who collaborate and work with us to share information, to share knowledge, because this is important not just for me, not just for my centre, not just for the University of Pittsburgh, not just actually for Pittsburgh. We should be proud that Pittsburgh can play a part. That's very important. And you referred to Salk and his legacy. There's a wonderful, I'm from Ireland, and we do giants in Ireland. And there's a wonderful term about standing on the shoulders of giants. You know this? No one person, no one place, no one virus stands in isolation from the rest of the world. We are a community. We trust, we need to communicate to our community, which is why it's super important for me to chat with you, chat with the Post, and we need to explain why we need to do this. But no one place will do it in isolation. But one thing which is absolutely certain, if I am given the resources, given the leadership, and put in Pittsburgh, one of the things, even though I'm just here for a short time, just over a year, one of the things that I have no choice to do is to play our part with our people in our place, and we will do our share of investigating how this disease spreads, how this disease grows, how this pathogen behaves.
Dr. Redfield from the Centers for Disease Control on Thursday made a statement saying that we have an understanding of how influenza, how flu works, how it attenuates, how it ends up being spread. We've got an idea then, as we're putting together viruses, it was putting together vaccines for it, how that is going. We don't necessarily have that same understanding for corona at this point. How long does it take you to get up to speed, to have an understanding, to then start thinking about making a vaccine?
So because it's a global effort and because people have lots of experience, so remember that this is not the first emerging infectious disease. We have had bird flu, 2009, which led to the death of 300,000 individuals globally. We've had the Ebola outbreaks in West Africa. So there is a collective knowledge in the community about what to do. But as new viruses emerge, and SARS is another great example. SARS is closely related to coronavirus. In fact, now this virus has the same name. It's basically SARS coronavirus 2. And that allows us to take the knowledge which was gleaned during the SARS outbreak and determine what types of vaccines would need to be made. What's really surprising is there are already nearly double the number of people who have died of this novel coronavirus than died of SARS in the complete outbreak. So it demands collaboration, it demands connection, and it also demands that we then begin to do some work. So what do we do? So there are numbers of different ways in which you can make a vaccine. In the olden times, in many olden times, a number of years ago, Salk, for example, in those days, they carried out a process called empirical attenuation. Okay, so that's technical. Empirical is basically just using your best judgment of what to do, and attenuation means weakening. So this virus, if it was the real virus, is the strong virus which causes the disease, which gives you pneumonia, which creates the condition known as COVID-19. This is what the disease is called. So what do we need to do to change that virus, which causes disease, into a virus which doesn't cause disease? In empirical weakening or attenuation, we take that virus and we grow it in inappropriate cells. So if the virus comes from a human, we can grow the virus in chicken cells. So we pass it from the human into a chicken cell, chicken cell, chicken cell. Over time, the virus adapts to grow in chicken cells and not
human cells.
Not human cells. And whenever we then get to the stage where we say, we think this virus has changed sufficiently, we have a model in an animal that we take that potential or candidate vaccine, we inject it, because we don't want to do this first in people, right?
Certainly.
Because remember, vaccines have to be safe before we put them anywhere near kids. So we take it, we put it, we vaccinate the animal, and then we give the animal the disease. And if the animal doesn't get the disease, we know we've got a vaccine. That's the old style of vaccine development, and there still is a part for that in this outbreak. But what we can also do is now, because we have all of these genetic tools, we can do things quickly. So we can take the genes from the virus. In fact, we can order the genes from the virus using a computer to a company that can make those genes, even if we don't even have the virus, and they can send us the genes, and we can plug those genes into other safe vaccine backbones, right? This is, so if you imagine we've got a really good vaccine,
right.
Measles. Measles is a brilliant vaccine. And we take a bit of the coronavirus and we put it into measles so that that genetically engineered recombinant virus looks like measles and also like the novel coronavirus. And we're able to grow it and we're able to perpetuate that virus. And then we're able to take that and put that into an animal. And if we look at the animal and say, does it make measles antibodies? Yes. Does it make coronavirus antibodies? Yes. Could this be a candidate vaccine? Yes. And then we take it further and then systematically we grow it and we ask the question, would it work in people? Which is what Salk did.
Just an absolutely fascinating explanation of some of the work that's going on, and I think that we could probably talk about this for hours and hours, and Jerry, just begin to get a test, an understanding of what's going on. Thank you for the explanation as to what's happening, and again, we will be in touch with you as this work continues to go forward, because I know there are going to be a lot of hours spent working on this. Thank you very much for being here.
Thank you very much.
Dr.
Pleasure
Paul
to
Dupre
talk to you.
From the University of Pittsburgh. And by the way, if you want to continue to follow him, Dr. Dupre is very active on social media, particularly on Twitter, at 10Qs. 10QUEUES, at 10Qs is his Twitter handle. And coming up, we will have much more on the KDPG Sunday edition. Even if you're looking for a job or you want to make certain that your voice is counted, a big project that's happening here in Pittsburgh, and we'll switch gears and talk census right after this.